information. So there, and cool. So we can go back one slide. There we go. Just as a reminder for everyone that's just joining us, please change your Zoom and Slack username so that you have your first and last name, and also most importantly your team name. This will help us to communicate and make it easier for discussion so that we know who are we talking to. And some housekeeping items here. Please don't forget to keep up your mic muted unless you're presenting or you're talking. Also, we would like you to have your cameras on. This is supposed to be a really casual meeting so that we can just get to know each other, talk a little bit more, present, have some fun. And finally, don't forget to raise your hand if you want to participate. Here we also have Ryan and Gabby. Ryan from UBCO and Gabby from ASU as well. They are going to help us in the background. If you need anything at all, please don't hesitate to contact us in the DMs. And one last thing, if you're not already in our Slack channel, in our Slack workspace, excuse me, there's going to be a link soon being posted in the comments. And yeah, we're going to talk more about it later. So again, thank you for coming.
Yeah, so before we get into our plans for this year's Phototroph Community, we want to tell you a little bit more about what the community is and how it came about. So to do this, we have a former member of the 2021 Phototroph Community and also from the grand prize winning Marburg team, Cedric. So he's here with us today to talk about how the Phototroph Community got started and also what they accomplished in 2021. So Cedric, whenever you're ready.
Thank you very much. I hope you can understand me. Okay, fine. Can I share my screen real quick? Okay. I hope this works. You can see. Okay, thank you very much for inviting me. It's really nice to be back here. So as I've said before, first of all, we will talk about the history of this workspace and of this community. Then we will go into our project for the 2021 season. And yeah, to close things up, I will talk about some tips and tricks and potential pitfalls you will face during the iGEM season. Okay, so first of all, why are we even working with phototrophic organisms? I mean, it's a bit like preaching to the choir. Like most of you are working, all of you are working with phototrophic organisms. We are in a phototrophic community, so I don't really have to talk about why we're using this. Of course, it can be many things. It can be addressing the climate crisis. It can be creating a more sustainable future for all of us, or some unique features your model organism can offer to bring to the SynBio community. But then this begs the question, why are there so few iGEM teams actually working with phototrophic organisms? In the 2021 season, we have looked at this for the last, back then it was 6 years, like how many teams are actually working with plants or with phototrophic organisms. And we could see that out of like almost 2,000 teams, barely 2% are actually working with plants. There are a few more for cyanobacteria, but it's still like a very, very tiny minority of teams working with phototrophic organisms. And as you've probably experienced, this is mostly due to like the typical problems while working with phototrophic organisms. They are hard to work with. They are slow, they have growth limitation, both in time and space. There is a severe lack of multiplexing, like you can't run that many experiments at the same time. And of course, there are missing tools. Some additional problems the iGEM teams have faced is the lack of support they have. Like most of the team we have asked don't really know their organisms they are currently working with, and even their PIs, instructors and advisors lack the knowledge. Additional problems we have experienced during the 2020, 2021, and even now is of course the COVID situation and therefore the isolation. You can't really exchange that much information with other teams. Other than that, there's of course also the lack of iGEM resources, which hopefully will change in the near future. But if you go to the registry pages and so on, you will see if you're working with phototrophic organisms, there's not that much resources at the moment. Even like the missing tools are like, we don't even have that many tools at our disposal at the moment. So this is really something that needs to be addressed in the near future. So this is the reason why we decided in the 2021 season, why we want to address this issue, bring teams closer together during the COVID season, and maybe troubleshoot some projects. So how did we actually address these issues? So first of all, of course, there's the Slack workspace, which you should join, of course. There you can exchange information, can talk to teams directly. It's really nice to have. Then there are the meetups. Back then, we decided to do a mini meetup series of 4 meetups, which retrospectively seems a bit too much. So it's a good decision that you decided to do less than 4 meetups. This was really way too much. We invited experts from the industry and academia for all of these sessions, and also did after the presentations were over, we had some troubleshooting sessions for different organism types, for example, for algae, for plants, and for cyanobacteria. And something which you should really take advantage of is the opportunity to connect with other teams and to get feedback, to seek some help if you need any, and to offer help if you can. Lastly, but most importantly, I think it's the Phototroph handbook we designed and created. It's a 160-page handbook created in collaboration with 11 other teams, which you can find on all of our wikis and hopefully soon also on the official iGEM pages. So this does not only contain directly to just the phototrophs themselves, not just the cultivation, but also modeling aspects and also some inspiration from past projects. Some really positive side effects we have seen after the collaboration was over was that there were multiple spin-off collaborations. So there were multiple teams, for example, in the cyanobacteria community, which decided to work together afterwards. There was like very positive feedback for the troubleshooting sessions, which helped a lot of teams. And some of the teams even connected with the experts afterwards. And this helped them fulfill their IHP, the Integrated Human Practice criteria. So I'm very excited to see what the future brings. As said before, seek help if you need help, help other teams, and get feedback on your project. So then let's move on to what we have done in 2021 for our own project, for the Open Plus project. We wanted to really have an impact in the plants and bio community. And of course, like one of the most challenging aspects we will face in the future, even if we act now, is the impact of climate change on agriculture. So as you can see here, we will most likely face a 70% harvest loss annually compared to now. We will face a 20% less arable land, and we will face water shortages. So we have to produce more with less space, with even more losses, with less water for more people, which is a really challenging thing. And we don't have that much time to address these issues. So our project was about like a foundational project about addressing those issues and having like a self-reprototyping system to engineer crops, because as you see, like 30 years is not that much time. You can't actually work with plants. You need a rapid prototyping platform to address these issues now, and then getting those insights into the actual plants themselves. So why did we decide on chloroplasts? I mean, it's kind of the obvious choice if you want to engineer plants, because this is actually where the CO2 fixation and the light capturing is happening. So for this reason, we decided on the chloroplast, but there are also many other very nice reasons. For example, you can do high precision engineering, which is not really possible at the moment for nuclear transformations. There's an absence of epigenetic silencing, which is also nice because it makes it way easier to engineer. And if you want to produce some product, for example, vaccines, there's also a high level of protein expression in the chloroplast. Also, one of the major reasons we decided on it was that chloroplasts are mainly inherited maternally, which means they are not transmitted through pollen, which is really nice to have as a security, as a biosecurity feature, because this is one of the main concerns people face while engineering GMOs. So how did we go about this? This is a really, really rough abstract. So what we have done is we have grown our own plants. Then we taken the biomaterial and extracted the intact chloroplast from them, and then lysed them to to just have this this rough transcription translation machinery itself without the cell membranes, without the structure. Afterwards, we've added the reaction mixture, so different salts, ATP, nucleotides, and so on, and then our own Golden Gate-based constructs to test. So we've done this whole system for tobacco, for a relevant crop, wheat, and oak and spinach. Coming to the achievements, okay, we have also tested rice, but rice wasn't that successful. It was still successful, but yeah. But it kind of shows like it wouldn't really be feasible to do an oak project, for example, or a wheat project during an iGEM season. This is only possible if you work with cell-free systems of those plants. Some other achievements is there were only 18 parts for the chloroplast at the moment we started our project, and none of them were actually tested in the chloroplast itself. So we added 157 novel parts, which most of them were also tested. Also, we've done the first chloroplast transformation in the iGEM community, which is a really time-consuming project. And yeah, for example, also what is probably good to mention is, for example, wheat is currently not transformable via chloroplast transformation, as we know of. So to actually test construct and to test metabolic pathway, it is not feasible in vivo at the moment. So you have to use an in vitro system like the cell-free systems. So this was very surprisingly very successful. So we've won, yeah, a shit ton of awards for it. And in the near future, or currently, we're working on a publication for this project. And I would also highly encourage every one of you, if you have some novel insights, if you are really hyped by your project, you should pursue a publication afterwards. It's really worth it. But coming to tips and tricks, the success was not always the case. So I've done iGEM for the last 3 years, 2019, 2020, and 2021. 2019 was kind of sad because we put a lot of work into our project, but because we didn't really understand the Miller criteria and we were under severe stress, we fucked up the gold medal criteria and only got silver. It got even worse 2020 when, after months of work put into the project, we had to pull out of the iGEM community due to COVID. So it maybe shows you, yeah, it's not always that happy. So my first tip for all of you, really take the medal criteria seriously. So you have to have like 1 or 2 people who keep checking and keeping track on the criteria itself. And you should have at least 1 backup for each and every single one of your criteria. So of course, like most importantly are the bronze medal criteria, secondly, the silver, and third, the gold. And for the gold medal criteria, you should not pursue all of them, but you should pursue at least 1 more. And of course, if you are unsure about it, please ask around. You can ask your advisors, you can ask other teams, and you could, of course, ask the judging committee itself. We have done this several times during the last year. Then, honesty. Honesty is really important for a project. Like, especially now that we are out of this like rose-tinted glasses phase of the competition and things get way more stressful, you have to be honest with your fellow team members. You have to communicate if you don't have enough time or what your time commitment actually is. If you have issues, for example, personal issues you are facing, or like if something doesn't work, just tell it straight out. Like, honesty is the backbone of your team. And of course, communication is key. You have to communicate with respect and with honesty. Lastly, a bit related to this is don't oversell your project. So there's a fine line between actually like selling your project, which you have to do during the iGEM season, and straight up lying. So most of the iGEM judges are professionals. They are PIs, they are postdocs. They have done iGEM before, so they will know about this, and yeah, you will hear about this. Something which gets kind of neglected during the iGEM season is teamwork. And this doesn't seem that important at first, but team building is probably one of the most important things you have to do. You have to have team building exercise. You have to do something together. Otherwise, your team will fall apart. You will have stress, and yeah, this is not to be underestimated, this issue. Also, most of you are currently working in sub-teams, subgroups, I would imagine. So really work towards building a cohesive project story. And of course, yeah, you will win and fall as a team. Don't be discouraged, because as you will see later on, like when people or when teams present their projects, like it will always be kind of astonishing, kind of, wow, they have accomplished all of this, and for us, this PCR isn't working, this mini prep isn't working, this such and such isn't working, and this is normal. Like all of the teams are facing this. You will only see the success story. You will only see a fraction of the work that is actually being done, and yeah, keep that in mind that not everything is working out. For example, for us, like we have begun the 2021 season really, really early, and we had some working extracts at the end of February, start of March, and then afterwards, for some reason, it didn't work out at all. Like for months on end, till the summer, it didn't work. So it is insane. It is a roller coaster, iGEM. And also, if you don't have results now, just keep working. Like this picture I'm showing you here is one of our team members, which was actually in the laboratory till 45 minutes before the wiki freeze. It was 5 o'clock in the morning for us. So really keep on working and don't let perfect be the enemy of good. Lastly, collaboration. You should, of course, like I'm mentioning this the third time now, but really take the collaboration seriously. Try to contact other teams, get feedback. This will really help you also with the judging session later on. Like if you don't get any feedback until the actual Giant Jamboree, you will have a severe disadvantage. You will need to get feedback from the expert, from other teams, and you will, also hopefully seek help if any help is needed and offer any help if you can offer any help. And lastly, but most importantly, have fun.
Thank you very much, Hendrik. That was really cool. So yeah, basically we wanted to bring you that, guys, so you'll be able to know what was this community about. And yeah, I wanted to let you know from all of us that we are really grateful that you guys have done all this together. It was really amazing. Thank you again. So continuing with that, basically we just wanted to let you know that what we did is that we wanted to bring this community, we wanted to continue it because we really thought it was a really good initiative. It's basically the opportunity for all of us to interact, and really there are not many people that are working with Phototrophs, and it's our opportunity to really try to demonstrate that community that there's people that are willing to work on this and that we can push it a little bit more because of all the importance, the climate importance, and because yeah, they're really cool. Yes, more organizations to work with. So team is ASU and UBCO partnered up this year to try to bring back this community, and we have some of their goals that we want to go through this that we're going to talk later. So we can go to the next one. And one of them is, for example, the Phototroph handbook that we were talking before. And we'll go more in depth later, but just to say something here, we would really like to update it. There are so many more things, and this is like a really good resource that last year was created for all of us to try to help the teams now and the teams that are going to come in the future. Mm-hmm.
We're also planning to have the first ever iGEM Phototrophs virtual conference, which will be an online event where all of the participating teams can present their projects with experts to gain really valuable feedback ahead of the giant jamboree, and also listen to talks given by representatives from industry and academia, kind of like how it was organized last year. And of course, we'll also provide a lot of opportunities for all of the teams in this community to discuss their projects with mentors and researchers along the way, whether that's at our next meetup or on the Slack channel anytime. And another great part of the Phototroph community is the ability for teams to earn badges based on the types of contributions that they've made to the community. So these badges will be sent out later in the year, probably around in September after Volume 2 of the handbook is complete, and can also be included as part of your team wiki and your project videos, or even on your posters if you're presenting on posters to display your contributions to the iGEM Phototroph community. And this way it makes it very easy for iGEM judges to see that you've fulfilled various parts of your integrated human practices requirements too. So these are actually the badges that were used last year. So yeah, like it says on the slide, the designs and the descriptions for them will be slightly updated based on how we're organizing it this year, but the main idea behind them is all the same. So the organizational badge is for teams who are involved in organizing the community. So that would be our team and the UBCO team. The presentation team, or the presentation badge, sorry, is for teams who choose to present at the Phototrophs virtual conference that we just mentioned earlier. The handbook badge will be for teams who contribute to writing Volume 2 of the Phototrophs Community Handbook. And then the community badge is, of course, for attending the meetups and all of our events and being part of the community. And then there will be one more additional badge that we're adding this year, the video badge, which will be for helping creating our video protocols, which we didn't really mention when we talked about the handbook, but another aspect, another angle of the handbook that we're going to be adding this year is that we'll be also pairing it with video protocols to make the experience of going through that handbook a little bit more interactive. Say you're reading about how to do a certain lab technique, but it's not really clicking when you're reading it. You can click on the hyperlink to the video that we will all be creating, and then you can watch someone walk through the steps of it there. So we really hope that you're looking forward to being part of this community as much as we are, even just through the brief description that we've had just now. But now we'll have René Inckemann, who is a PhD student at the Max Planck Institute, where he researches phototrophic organisms like the microalgae Chlamydomonas reinhardtii and just chloroplasts in bio generally. He's also representative from iGEM's Plant Subcommittee and was also an instructor for the 2021 Marburg team. So today he'll be talking to us about what the Plant Subcommittee is and how they can be helpful to your team this year. René, whenever you're ready.
Thank you for that very kind introduction. I will try to share my screen, and I hope you can see it. Perfect. So thank you for the invitation, and for now it's amazing to see that like everything is coming together again, and that this community is being continued. Yeah, as already introduced, I'm talking now out of the perspective of the Plant Subcommittee, but like I have to mention before maybe that like Plant does not mean that we just talk about like just plants, but this is basically a Phototroph community, iGEM community, also about algae and cyanobacteria, right? So it's not exclusive or excluding any other phototrophic chassis. So I try to give you an idea what the Plant Committee is about, how it has been in the past. Then I also wanted to mention some like previous teams just to show like what is possible, and that like plant teams don't need to, or like phototrophic teams don't need to hide behind like any other teams. Then I would like to talk about like what our committee can offer now, and in the last part I would like to talk what we would like to offer in the future, and there I will also do some questionnaires here, or like some like interactive part I would like to see to get some feedback what we might need to, or what we might want to include in the future. So initially this committee, or like the Plant Committee, was founded in 2016, and they have done great work back then, and they came up with the iGEM Plant Prize, which I will talk about later. They came up with the first resources on plant engineering, and also some plant-specific parts. But after 2016, this committee already fall apart again, and then there was unfortunately no further activities. This initial initiative was also connected to the Open Plant Initiative, where many of the Open Plant Committee members were a member of. And this is just something I want to mention here. This is a initiative which wants to accelerate the development of tools and share them openly. And maybe you want to have a look on this website to see what they offer already and like into that program. This is the Phytobrick standard I was talking about, and this is also what the Plant Committee back then came up with. And this is about like a type 2S based cloning standard or Golden Gate based standard, which is about like having the same standard for all phototrophic chassis. So like every cyanobacterial type 2S, clammy type 2S, any plant type 2S, they all work with that standard. And if you want to have more information, you can also scan this QR code here. The website is looking very old, and we hope to update that soon. But basically, what you need to know from that slide here is there is a certain cloning standard which the Phototroph community is using. Okay, so another reason why I think, additional to the reason Cedric already mentioned before, why it's cool to work on a plant project or like a Phototroph project in iGEM is that there is actually a special award for like any other chassis or any other chassis don't have an award, but like if you work with algae, cyanobacteria or plants, you can get the best plant synthetic biology prize. And that's what especially all of you here in the call should definitely work on or aim for. And yeah, before there was not so much competition for this award, but like over the years now, I think there will be more competition for this specific award. Okay, so where's this new committee or like subcommittee coming from? So I was previously a part of the engineering committee in iGEM, and this was not focusing on plants. And then I was looking at like the old initiative of the plant committee, and I was like a little bit sad that like there was no specific or efforts anymore to help like phototrophic teams. And this committee was before involved in like organizing this interlab study. I don't know if some teams of you here are participating this year, which is basically an experiment in E. coli where like every lab in the world or every iGEM team in the world does the same experiment. And then you compare the data in the end and try to like standardize until you get like comparable data. This engineering committee also has some educational material and videos and webinars out there. So like if you struggle with cloning or with anything in iGEM, also for this year specific changes, there is like some material if you want to look into it. But now basically coming to this plant committee or subcommittee, we started in the beginning of the year with the goal that we want to help teams working with photosynthetic chassis to have a better start, because like we felt that the entry barrier to work with a plant with this clammy is way higher than like if you would start a team with just E. coli or yeast or something like that. So therefore, the goal would be to lower the entry barrier, and we try to identify what are the entry barriers, and we identified some, but maybe in the end of the talk you can tell me what other entry barriers you see for working with a photosynthetic chassis. So now I want to come to like some example projects to tell like some success stories of previous iGEM teams, and this is one from Valencia from 2017. And what they basically did is they wanted to have like a communication channel with plants that you can basically talk or like turn on and turn off plants. And what they did was installing genetic light switches into the roots, and then they could like switch on and off like certain gene expression and change the behavior of the plant by that, which I found quite cool back then. Another project which is not like working with higher plants is the Kaiserslautern project from 2019, and they were second runner up. So like in the finals, there was a climate project in the finals, I want to remind here. And what they did was basically installing plastic degradation into clammy, and then trying to like get plastic out of the water with that engineered clammy strains. And they were quite successful and could show that they can degrade PET with clammy. And then you don't need, if you compare to E. coli, you wouldn't need to supply any food for the microbes to grow, but clammy can just grow on light, right? Another example from last year, who also in the finals in the undergrad section, is the Toulouse team, and what they did was basically combining phototrophic organism with heterotrophic organism in one culture, and then having a perfume production in the heterotroph host. So basically, the cyanobacteria were secreting sucrose and were engineered to provide the food for the yeast. So the whole culture as a whole was basically without any feedstock, and then producing perfume from just CO2 basically in total. And they were also like grand prize, I think it was grand prize in the undergrad section. So as you can see, there's already Cedric already presented the Open Plus project, so I don't need to mention this here. So like there's quite a hype, or like quite some very recent success stories of phototrophic teams. So maybe one of you here in the call can continue the success story for this year. Okay, so now coming to some resources, and there I definitely know that the resources now on the iGEM web pages, or especially on the old iGEM web pages, are not very updated and are not very good. But I still wanted to give the link here, so because like these links are really hard to find. And this is basically a website where like every plant project until 2017 or so is listed on this collection page, and there's also some like tips for parts. But like this is not what we plan to remain on, or like rely on in the future for the plant committee. Now we are developing, and this is just like almost empty website. We are now moving to a new website, and there will be a plant, or maybe we will also just call it Phototroph synthetic biology page. And the plan is to put a lot of resources on that new website. But we, as I said, we just started in the beginning of the year, and we are not many people yet. And the idea would be to collect as many resources as possible on this new website. And I hope by the end of the season, you can still come back here if you now go to that website, or like if you now save the website and find some more resources. And I also hope that I can promise you here that we will try to put your Phototroph handbook on that webpage, that this is not just displayed on one of your wikis, which is also great, of course, but like I hope that we can put like the handbook just directly on the iGEM main page here. Okay, so what did we already achieve? For now, I just talked about like what doesn't work at the moment. So the distribution for this year shrink down a lot because of technical reasons. And in the beginning of the year, when I asked the organizers, is there any single plant part on the distribution, they were like, not a single. Is there any clammy part? Not a single. So then I got 3 days, or our team of the plant committee got 3 days to include something. So we focused just on the 20 most important parts to engineer plant chassis. For now, it's rather for Arabidopsis and tobacco and stuff like that. But in the future, we definitely want to include cyanobacterial parts and clammy parts and so on. So if you would have feedback there, what you would like to see in such a distribution, or like in a plant collection, which is sent out around the world, that would be very helpful, because of course we can come up with plans what to send out, but maybe you have completely different ideas and suggestions. Something we are also very heavily working on is a list of protocols which are not so difficult as already mentioned, as like a typical paper protocol, because it's super hard to follow, for example, just the normal plant transformation just from a paper. But what we try now is basically having like pictures and comments on the pictures, like what to take and what not to take, and basically having a more interactive protocol. But like I love the idea of having videos instead of having instead of relying on like just text and pictures. So videos would be the dream of even of us to like provide to the teams, to have like for every main photosynthetic chassis, like the basic protocols in a video format. That would be amazing. Okay, what we also already did this year was basically meeting up with actually quite a number of iGEM teams from this year to give them individual feedback on their projects and help them during the season. And we haven't reached to every iGEM team. So like if you know any other iGEM team which needs help, or if someone is here who needs like just a individual Zoom call to discuss very specific things, we are a network or like a group of experts which have some experience with different chassis, with different techniques. So you can always reach out to us if you need help. Okay, now we come to more the idea or the more broader vision of this plant committee, what we would like to establish in the next years. And this is, for example, about setting standards, because we feel that this is even harder to achieve compared to other chassis like E. coli, because like there's so much more to take into account. So for example, in my previous research, and also like some iGEM teams have already shown that like the light quality, the light intensity is affecting heavily the quantitative experimental outcomes of your work. So like if you don't do standardization on like how much light is if getting on your cultures or on your plants, and like what is kind of like light source do you have, this can be really hard to standardize. And something like that, like not just for light, but like for every aspect of like phototroph cultivation, would be really nice to standardize. And then of course there's not many enough characterized parts in the distribution. There we need to like change a lot to have like a better basis for like starting with a Phototroph project. Then we have identified 3 major things as resources which we want to provide. That's basically a database for a lot of Phototroph things. So for example, sorry, now I have a blackout. So what are the specific conditions, like protocol database and like strain database and everything? And having that in one spot on one website would be amazing, I think. Then we heard back from some teams, especially in South America, that like getting seeds at some universities from a certain Arabidopsis or like tobacco, whatever plant is really hard. And this is something we haven't thought about in the US and in Europe. But like having basically an address or like a email address to write to if you want to have like a certain strain or like a certain seed, a starting seed for your project, would be something that we have like a hub or a network of like addresses where you can very easy and fast get these strains. The same goes for agrobacterium to transform plants. This goes for clammy strains. I mean, for clammy there is a good address to other things, but like we still want to have like some helping hands here. And then of course, as mentioned before 2 times already, the parts collection is one of the keys for like more easy starts for the teams. And then one of the most ambitious projects which we have in mind for the future is doing interlab. People are now doing with E. coli, but with phototrophic chassis. So This can be clammy, this can be tobacco and Arabidopsis, this can be cyanobacteria, and then doing a simple gene expression experiment and see if we can compare data around the world. Because like this is already hard in E. coli to just measure GFP in different labs and then get the same thing out of your data. But we think, especially for plants and for clammy, that this will be even harder to standardize. And like running a first interlab to see what the problems are, that would be amazing. So, and I would like to hear your opinion on that later. So now we come to the more interactive part, or like to your questions, and there you can either type the answers into the chat, or you can also probably raise your hand and talk if you want. It's just 4 questions, so I don't do like 100 questions now here. So what kind of resources would you like to see from the iGEM Plant Committee? Is there anything which I haven't mentioned, or like something more specific, what you would like to have, or what you think could be a huge help for like future teams to start with, or even for your team now? Is there anything? I just wait a few seconds, otherwise I continue. Monocot parts, that's a really, really good point. So like, that's true. We haven't included monocot parts yet. And that's a really good point. I will write down on my list. But the reason why we haven't focused on them for now was like, yeah, there's not many teams. I think there's not a single team in the iGEM history which has worked for the for monocots. So if your team is actually working with a monocot, chapeau. And I really cross my fingers for you, because that would be amazing alone that you are working on maize or something like that. But that would be actually the dream, that like teams can work with maize in iGEM. Yeah, that's also a good idea. So I think I will copy the I'm not so fast. Yeah, chassis organization. Yeah, cool. Yes, cool ideas. Okay, I think then I will go to the next question. If not, you can always write us these suggestions via mail in the end, or like tell me in the end. The next question is, what kind of path would you like to have? Is there, yeah, and this is already answered. This was already answered in the previous question. But is there anything specific besides like regulatory parts, or is there anything which comes to your mind which would be quite useful for many teams? Is there more? Chromosomal integration. Good point. Yeah, we haven't done that yet. Probably you are also talking about cyanobacteria, right? Yeah, and I think there's no chromosomal integration part in the complete. I don't. Yeah, there's not many chromosomal integration parts in the iGEM registry, and if so, they're probably not characterized. That's good. Yeah, cool. Very cool that I get some feedback here. Okay, let's continue with question 3. What is your opinion on a Phototroph interlab? Is this too much work for iGEM teams? Is there too much work for iGEM teams to do another experiment if, like, for example, the constructs are already provided? The idea would be, for example, we send out 3 constructs for that chassis, and the team would need to do the transformation and then measure something. Maybe this is asking too much. We were thinking the same, but on the other hand, we would then integrate it into the medal system or something like that. That like teams can tick a medal box or something like that for that. Is that asking too much, or is this a cool idea, or what's your opinion on that? Yeah, the reward is insane. That's true. Yeah, Yeah, agree on Gabriela's comment with the detailed instruction. Yeah. The other point which I had in mind for this was like in the previous iGEM interlabs have all been published and like all teams are authors on that paper. Yeah, this was slow to start. Totally, I can totally understand. But like if like for example we could offer like a 100% chance that all the team members who are actually doing this interlab can get a publication, that would be probably also then playing into that equation if this is still worth to invest, right? And we were thinking already contacting a journal before if they would accept like such a paper, and then yeah, try to like do this experiment and then put all these everyone on that paper. And I think I get some cool feedback here. Cool. Thanks for the feedback. Okay, and my last question I think is, do you have like any other ambitious project idea, what a plan of Phototroph community could offer to the teams or could do in the future? Like it can be as crazy as you want. But we are just collecting ideas here on things to make iGEM Phototroph community a better place. Any ideas? If not, if you have some, you can always write this to me via mail. And with that, I'm happy to take any questions. And here's the email address again: engineering plans@igem.org, and this will go to all of our members, and then we will hopefully answer you. If you don't get an answer, don't hesitate at all to just send the email again. You don't have to be ashamed, or like sometimes just emails, yeah, also in my email program get lost or something. So don't hesitate to send them 2 times or something. Yeah. More Photoshop strain banks. Host workshops and workshops. Yes, cool. Workshops we were also thinking of. Yeah. Workshops, strain bank. Oh, this materials loan programs, Gabriela, is amazing idea. I don't know how to realize that, but I know many teams who have actually that problem. Yeah. Yeah. So because we also realized that like the growing capabilities are also sometimes like the limiting point. And if you would either have like a manual how to set up like a Phototroph growth setup for like $100 or something. Okay, so like equipment. That's the kind of feedback I want to hear. Yeah, perfect. Thank you so much. And now I'm happy to still take questions. And oh, that's also cool. Yeah, a database for material from phototrophic teams. Yes, cool. Yeah. Thank you so much for all your ideas. Is there any specific question?
Do you think these resources that are currently available are underutilized?
From our committee or what?
For the Plant Committee.
Yeah, so like I think yes. I wanted to talk around it, but yes. So I mean, like we're also not many people yet, but we will next week we will make a call for new people for this Plant Committee, and then we will try to grow it by the next year to realize more ideas and to help many more people, because there's also getting more and more Photoshop teams, but definitely the resources are not completely used yet. Yeah. Cool. And then I'm still very excited to hear the.
One more
Yeah,
question.
sorry.
Is
Yeah.
there anything that you think would be helpful for us to include in our handbook this year that might like help co-achieve some of the goals of the subcommittee?
That's a very good question. Maybe something like what I was already thinking about, like having a instruction or a manual how to improvise with like low tech a growth setup, something like that. What is the improvised setup for cyanobacteria, for clammy, and for plants? I don't know if this is feasible or too crazy, but I have a PI who told me about $300 plant setup, which is on the level for publication. So it's still a lot of money for some teams, but like one can still think about something like that to include. Yeah. Other than that, I probably need to think about it a little bit more. But yeah.
Yeah, definitely. We'll keep that in mind then when we
Cool,
make
thanks.
ours.
Cool. So if anyone has any more questions, you had the email there. Are there any other ways that they can reach out to you, René, or the Plant Committee?
Yeah, exactly, with the email. Or what do you mean?
Cool. We'll proceed then.
Okay. Well, thank you so much, René, for speaking and giving us all of that great information and letting us kind of give you some of our ideas as well. So we hope that for all of the teams attending that was a helpful guide to learning some more about the Plant Subcommittee and what value that they bring to all of your teams throughout the competition. So now I think it would be a good idea to get all of the teams kind of talking amongst themselves a little bit more, and so we can get to know each other. So what we're going to do now is we're going to break you all off into breakout rooms with your other team members for around 5 minutes to regroup and prepare your answers to the icebreaker questions that we have on our Miro board, which the link should be in the chat soon. And also, we'll come back after that and present our project pitches. So during those 5 minutes, if you also want to talk to your team about who's presenting what or something like that, definitely feel free to. So I think, yeah, if anyone has any other questions, you can stay behind in the main room, but we're going to be splitting you off into breakout rooms now.
Yeah, and just to carry on with that a bit, well, you're getting there. If anyone's in here, we're going to use Miro. If you have any questions at all, please let us know. You can send us some DMs there. Thanks. So without further ado, just a summary of that, this document that we were working on is a living project. So keep updating it, have some fun with it, and yeah. And right now, what we're going to do is that we're going to give you each of you teams some time to present a little bit about yourself. What's your team about? Let us know everything about you. Be kind of brief about it. Use the Slack channel to let us know more about you in terms of personal information, and then about your project. So yeah, we can proceed. So team ASU.
Okay. Hello, my name is Gabby, and I'm the co-captain of the ASU iGEM team, and I'm going to present a little bit more about our project this year, which is called Chlamydomonas, and it's a bioremediation platform based on the chassis Chlamydomonas reinhardtii, which is a microalgae. Our team is composed of 11 student team members, and we're all split into various sub-teams. Some of us work on the wet lab and human practices and dry lab as in a standard iGEM team, but our iGEM team also has other sub-teams within it, such as our filter prototyping team, which I'll talk about later. We also have 4 faculty members from our university. Dr. Kevin Redding and Dr. Taylor Weiss have expertise in algae and phototrophic organisms, while our mentors, Dr. Benjamin Bartel and Dr. Christopher Plaisir, do not have direct expertise with phototrophic organisms, but they do have a background in SynBio and are able to help us with that aspect of the project. And also we have a few advisors who have been instrumental to our progress this year. René, who you heard speak earlier, has helped us a lot throughout the competition, and we probably couldn't have gotten to his point without his help. And also Emma Lieberman and Maggie Cook, which were the co-captains of our team last year, have helped us a lot, learn a lot of skills necessary to work with our chassis. So our project, what is our project all about? Our project is about engineering the microalgae C. reinhardtii to bioaccumulate arsenic from contaminated water. So in our state, in Arizona in the US, there's an issue with arsenic contamination in water in rural areas in and around the Navajo Nation. And we wanted to tackle this problem with SynBio by engineering this microalgae to take up or bioremediate this arsenic out of water. We're doing this by integrating 2 genes into this algae, ACR2P and PCS. ACR2P is arsenate reductase, PCS is phytokeratin synthase, but instead of worrying about those gene names, I'll just walk you through the pathway super quickly so you understand what's going on. So in the water, there's arsenic, and arsenic exists in 2 oxidation states: arsenate, which is the 5+ oxidation state of arsenic, and arsenite, which is the 3+ oxidation state of arsenic. These arsenic oxidation states or types of arsenic can come into the cell, and both of these states of oxidation states of arsenic will bind to phytokeratins, but in order for arsenic to bind to phytokeratins, it has to be in the 3+ oxidation state. So that's why we implemented the arsenate reductase gene into Chlamydomonas. So any arsenate will get reduced to arsenite, enabling it to bind to the phytokeratins. So basically, in a nutshell, the big idea of our project is that we're taking arsenic in the cell, reducing it all to arsenite, binding it to this peptide, which will bind to the arsenite, and then taking the arsenite up into the vacuole. So by taking up the arsenite and the phytokeratins into the vacuole, we effectively can sequester arsenic from water and decontaminate water. So this is what happens, and basically we're just taking up and sequestering this arsenic phytokeratin complex inside of the cell. So to do this, we have created a few different constructs. So this is our construct, which contains the 2 genes of interest, which I just talked about. You can see we have our transcriptional unit here with some basic aspects such as promoters, terminators, which we all need to express our genes. But then we also do have 2A peptides in between our genes to enable each of our proteins to be expressed individually within the cell. But the thing we noticed when we were building this construct is that engineering of multiple genes into Chlamydomonas reinhardtii, our chassis, is still in its infancy. And so that's why we wanted to add another element to our project to advance the SynBio community and the Chlamydomonas engineering community. So currently, there only exists through the literature a few 2A peptides which are known to work. So that's why, as part of our project, as sort of a side goal, we wanted to investigate 2A peptides and wanted to characterize many 2A peptides to see how we could advance engineering in Chlamydomonas. So very briefly, we designed this plasmid to test the efficacy of 2A peptides, which again are just small elements which, when a ribosome comes to translate, it induces a ribosomal skip event, which allows individual proteins to be produced through creating individual transcripts. And so this is the plasmid we designed, which has 2A peptides integrated in it. And the idea is that we have multiple iterations of this single plasmid, and each time we switch out the 2A peptide, and by characterizing the efficacy through a microscopy technique called FRET, we can sort of determine how well these 2A peptides work. And by studying the different efficacies of these 2A peptides, we can sort of come at the end of our project and say, okay, well, these 2A peptides work best in clammy, so any teams wanting to integrate multiple genes into this chassis in the future should use these 2A peptides over these ones, and that way we can sort of advance multi-transgene expression within clammy in the future. But in addition to creating these constructs and creating this system in the wet lab, we also wanted to propose an implementation for our project, which is our filtration system. So our filtration system is a system which we would ideally propose to integrate our algae into the real world so that our solution can actually be practical. And for this project, we are making a high-tech solution to our problem, which is high in cost, and also a low-tech solution, which is low in cost, so that we have two different solutions to present to the iGEM community to sort of show that our Chlamydomonas system can be integrated into the real world.
Cool. That was nice. Just to something that I forgot, if you go to the Miro board, you can also add your comments or questions and as post-its or sticky notes throughout all the presentations. So have that in mind. That'll be really useful for all of us. Yeah. Next up, actually, is us. So Ryan, let's go.
Okay. Hi guys. I haven't introduced myself yet, but I am Ryan. I am a member of the UBCO iGEM team, specifically working in the wet lab and human practices. And Gustavo and I are going to talk a little bit about our project and what we're all about. So before we go over a presentation of what our project actually is, here are a few of our members. This is our first time competing in iGEM as an iGEM team. So we've had to build everything up from the ground up, but we're super excited. And we're also, of course, super excited to be able to contribute to this Phototroph community. But anyways, to get back to what our actual project is about, our project is Lifebulb. And if I can just second.
There you go.
Yeah, sorry about that.
Cool. So yeah, we can say a little bit about what we're doing. So this year we decided to go for this project, which is Lifebulb. We are working with Synecocystis, so we are a cyanobacterial team. You can go next. Basically, our idea was that some some logo thing that we made. So basically, our idea is because we were really passionate about what is bioluminescence. We need light for everything, so light has been used for a long time, and we need it literally for everything we're doing, for extending the time that we are able to do stuff, and also for recreational purposes. Right now, the solutions that we have out there are not very varied. We can go continue. And we wanted to see how can biology can help us that in a certain way. Biology is able to do this with bioluminescence. Basically, it's taking the substrate, which is luciferin, and the enzyme luciferase, putting them together and producing light as a reaction. So again, we use light for a lot of things. Right now, our best solution are LEDs, but they are far from perfect. Lighting still accounts for 15% of the global energy consumption, and 5% of the greenhouse gases are produced because of this sector. People are trying to push for transition entirely to LEDs because they are the most sustainable thing that we have, but they are still have some carbon problems in that. And there's even more problems actually. So apart from the climate crisis, we can go to the next one. We also have the problem that we've gotten so good at producing light. LEDs are so cheap that now we have the problem of light pollution. basically this means that there's so much light out there that this is having problems in wildlife, is affecting them, and also human health. Stargazing in some places is even really difficult to do, and we are literally being disconnected of nature at this point. So yeah, again, what we try to do here is to try to envision a world that can be illuminated with life. Imagine a light that can grow by itself, a light that can be scaled up, something that can produce a dim light that is just enough to make us look at night. What else? Let's make it even greener, but make it be energized by the sun. And that's how we came to our project. So what we're doing here is that we are engineering the cyanobacteria in ecosystems. So next. With our project with LifeBall. We are engineering the Synecocystis sp PCC 6803. We are adding fungal genes that we're going to talk about later with the hope of producing bioluminescence. So I think, Ryan, you can take this one.
Yeah, so the pathway that we're expressing in cyanobacteria, it converts caffeic acid, oh, sorry, wrong slide. It converts caffeic acid into 3-hydroxyhispidine, which is then oxidized in a light-emitting reaction and recycled back into caffeic acid. Although cyanobacteria don't naturally produce caffeic acid, previous studies have successfully expressed caffeic acid production in Synecocystis, enabling us to produce carbon-negative light without the addition of external substrates. And we're also planning on participating in the iGEM 2022 Interlab study, performing mutagenesis to alter the wavelength or quantity of light produced, using circadian promoters to express bioluminescence at specific times a day, that sort of thing, to try and explore different ways that we can use bioluminescence and use synthetic biology to produce light and solve some lighting problems that we as a society face. So bioluminescent cyanobacteria possess a wide range of applications beyond just traditional lighting. Bioluminescence serves as a unique, promising, and profound medium for artistic expression. So bioluminescent art and design has an unparalleled ability to sustainably convey the beauty of the natural world that is especially relevant within the art and culture of Indigenous Canadians and local Indigenous artists. Our project demonstrates that synthetic biology can be more than just practical, it can be beautiful as well. By demonstrating what beautiful things SynBio is capable of creating, we hope to improve public perception in the field and reduce the stigma surrounding GMOs and genetic engineering.
Cool. And as you all know, we wanted to bring the Phototroph community back because we truly believe that the future of synthetic biology relies in the phototrophs. We want to make a difference in the climate crisis. Yeah, thanks to all of you for being here as well.
And
No.
Beyond the Phototroph community, we're also in the process of contacting a variety of local and international experts regarding our project's possible applications. This includes local Indigenous professors and artists, representatives from biotech companies related to our project, and experts in the lighting industry. And to further our goal of improving SynBio's public perception, we produced a survey to assess opinions on genetic engineering. And the feedback from that survey will help inform us on how we can use our project to reduce negative perceptions of GMOs. And another aspect of our human practices related to this perception of GMOs and synthetic biology is a series of workshops that we're conducting with local children. And these workshops involve interactive activities that can educate children by demonstrating the possibilities of synthetic biology and the wonders of science. And here are a few images from these workshops. We're still in the process of conducting them. They're a ton of fun, and they're great for spreading the word about what synthetic biology and science can do.
Cool.
So
yes, some approaches that we have here in the dry lab. We are also trying to think how are we going to put these organisms into the real world. We are thinking both in big and small scale. We are taking inspiration from some bioreactors that people have created to produce cyanobacteria inside of homes, so that you are able to use this as a biological lamp. Yeah, let's keep going. Something else that we are taking into account is the phenomenon of cell shading. All of us that we are working with phototrophs, the microorganisms, You probably have this problem. Basically, what we need to do is to try to model it, how's the best way of growing them so that the vessel that we are creating is able to uptake the most amount of light and also being able to produce the most amount of light, so that is quite useful. Keep going. Cool. So we just wanted to say thank you for everything. And one more thing to add to this is that I don't know what happened with the slide, but some of the problems that we want to tackle, maybe you can find it in the canvas. Some of the problems that we want to tackle is that we want to make this system even more efficient. So we are going to try to make different constructs that have different promoters, different orders of these ones. We also have some different genes from different fungi that have not been tried before. We are our hopes is to make this project so that we are, again, trying to provide a solution for the problem of of biotechnology that we are all facing. We also want to try to make this a project that will hopefully provide a new way of producing light in a literally biological and in many ways green way. And of course, we've seen that there are many ways of improving this because of mutagenesis that we can do. And there's a huge way that we can put it into the wildlife impact, how it affects our psychology, and of course, the climate crisis. Thank you. Cool. Next up, we have iGEM Thesali. Just a reminder that you can always comment and put your questions in our Miro board.
Hi, everyone. My name is Eli Maguta, and I am the leader of the iGEM Thesali 2022 team. Today's brief presentation will include the description of the phenomenon we are facing and how our idea contributes to its solution. This is our team. Here you can see all the members of the team. So this year we are trying to solve the environmental phenomenon called eutrophication. But what exactly is it? Eutrophication refers to the phenomenon in which excess nitrogen and phosphorus and other inorganic nutrients enter a water body, such as a river, a lake, or a reservoir. Eventually, cyanobacteria overproliferate in the water, leading to the consumption of the dissolved oxygen and the production of chlorophyll A and toxins, resulting in water quality degradation and the death of fish and other aquatic animals. Consequently, biodiversity, which is of great importance for the balanced functioning of the ecosystem, is reduced. The project Navanthus, a name derived from the fusion of Greek words navs, meaning sheep, and anthos, meaning plant, comes to provide a solution to this environmental challenge. Our team is participating in this year's competition with a water monitoring and bioremediation system in order to protect the biodiversity of aquatic ecosystem from eutrophication. Initially, our detection system, the black box that you can see in the picture, using a chlorophyll sensor and a GPS, will monitor the operating water bodies. Real-time data will update us on the surface's ecological status and, of course, on the eutrophication levels. Wherever our data indicate critical levels of eutrophication, a floating platform made of environmentally friendly materials will be placed in the water. The platform will be carrying genetically engineered plants that bioremediate the water body through phosphorus absorption. So this year we're dealing with plants in bio, of course. The plant that we will use is the common reed. The scientific name is Phragmites australis. The design we want to achieve in plant roots is to detect the presence of microcysteines in the water. Microcysteines are cyanotoxins And the detection will be done by a molecular riboswitch. The detection of toxins will activate the expression of phosphorus transporters in roots, leading to the uptake and storage of phosphorus, which is necessary for the plant growth. To be more specific, the detection of microcystin will be done by a riboswitch with a specific aptamer that recognizes the toxin. When microcystin binds to the riboswitch, the expression of tetar protein that is linked with the riboswitch in pre-mRNA form will be repressed. Then the expression of the next part of our system, which will be a PI transporter from the PHT1 protein family, will be increased, and the plant will uptake phosphorus from the roots by remediating in that way the waters. So our mission is to protect the aquatic resources in order to ensure equitable benefits for communities. The success of Sustainable Development Goals rests to a large extent on effective monitoring and bioremediation of waters and species protection, of course, without harming essential ecosystems. Thank you very much for your attention, and if you have any questions, we are at your disposal.
Thank you very much, iGEM Sally. That was really cool. Cool. So next on, we have HKU Hong Kong.
Okay, allow me to share my screen quickly. Yes, good evening from Hong Kong. Oh, good morning. Before I introduce our project, allow me to lay the groundwork of why we're doing it. So obviously, pathogenic infections have always been a thorn in humanity's side for quite some time. And it was only relatively recently that we were able to finally have a weapon against them, thanks to Sir Fleming and his discovery of penicillin. But obviously, the discovery of antibiotics not only revolutionized health, but also revolutionized industries like agriculture, pisciculture. But our reliance on these antibiotics have led to such a prevalent indiscriminate use that it's accelerating the emergence of antimicrobial resistant superbugs. Now the public health and industries that were once protected by these antibiotics are now at a larger risk from these superbugs. So there are many alternatives to handling this issue, alternatives that come all sorts of antimicrobial molecules. The one that we're going to be focusing on, and the focal point of our project, is antimicrobial peptides, AMPs, which brings us to our own initiative, AMPT. So the AMPT initiative revolves around transgenic AMP-expressing microalgae that is able to deliver passive immunization as well as nutrition to fish in aqua systems. The way we're doing this is through obviously our chassis of choice, which is a microalgae. It is Chlorella autotrophica, a variant of Chlorella vulgaris, known for its high productivity and high resilience. And we will be expressing the AMP cycropin B, which is a very quite strong antimicrobial peptide that is from silk moths, basically. So the AMP the AMP project is a multifaceted approach comprised of the initial 3 phases. Phase 1 is an in vitro pipeline for AMP expression and validation. This incorporates the high expression system of the microalgae with a robust and simple transformation and expression methodology. It focuses on the secretion of the AMPs and the detection of AMPs initially. These detection is facilitated by mCherry XL, which is an enhanced version of the well-known RFP mCherry, of course. The next part of this is after the secretion, we will be isolating this with a SUMO tag and SUMO cleavage, and then the purification. This allows us to get a very pure AMP product that is that we'll be using for in vitro analysis and validation to see if the AMP really does work, first of all. Phase 2 is the encapsulation. So AMPs will be produced and accumulated internally, and they will be conjugated to SUMO in order to increase longevity. So encapsulation is, will rely on 2 inserts, which will both be transformed into the microalgae. The first insert is obviously the expression of the sumo amp fusion protein, which is a quite simple constitutively expressed insert. And the second one is a nitrogen starvation induced promoter that is endogenously found within chlorella, which expresses the sumo protease. So the way this works is that once the sumo amp fusion protein is expressed in high enough quantities, we will begin the activation process, which is the expression of the sumo protease to cleave sumo, activating the amp for use. But because of the tiny nature of the peptide, it's very easily degraded. So after this, we will encapsulate and preserve the amps by freeze drying them. This will allow us to not only preserve the integrity of the amps, but it will allow us to provide aqua systems with a freeze dried product that they can use to feed fish and sustain the nutrient needs of these aquacultures. Finally, phase 3 is the adaptation process. So the versatility of our system allows us to work on novel peptides. So obviously, an easy place to jump to would be other amps like lactoferricin or ursocotaxin, which is a very another strong amp. But with this, a process can also be adapted to things like growth hormones, and it's definitely something we've looked at, but maybe is something we might not have time to explore. But something we do and we are exploring right now is the adaptation to various chassis. So currently in our labs, we have 2 strains of picochlorm, very, very high productivity microalgae, very, very high efficiency. And we'll hope to be able to adapt our purification pipeline and secretion pipeline to more chassis in the future. And additionally, to flesh out our system, we'll be doing quite a lot of modeling. So we'll be modeling the growth kinetics to determine whether or not the heterologous protein expression does have an effect on growth, positive or negative. Then we'll be also modeling the secretion model and see whether or not we can adapt this to more biosynthetic applications. Furthermore, we'll be doing in silico testing to simulate the real world conditions to identify as many possible outcomes as needed. Our team is also working on several relatively cheap bioreactors that we can use to not only scale up in production for lower budget applications, but due to the nature of the scale up, it is quite easily scaled up to industrial scale because it relies on a modular component. The details of this have not yet fully been finalized, but it's still something we are hoping to work on in the future. And that's basically the gist of our project. So if you have any questions, please feel free to put a note and then we'll get to it as soon as we can.
Awesome, thank you. I really like that you guys also mentioned your modeling efforts too. I think that was really interesting to hear about. And also, yeah, your slide deck was so pretty. Up next, we have UBC iGEM.
All right, that sounds good. Thanks, everyone. I'm just going to share my screen. Sorry. Okay, so just to give a brief overview of our project today, we're focusing on developing a heat-tolerant, designing a heat-tolerant crop for the changing climate. And to give a brief overview of our team, we are a group of about 16 undergraduate students coming from various different faculties and majors, from microbiology and immunology to biomedical engineering and biochemistry. So we have a wide range of undergraduate students coming, as well as many different graduate advisors also in wet lab or in dry lab as well. And our project this year is named Senestivum, and we're focusing on wheat.
And the main motivation behind our project was the extreme heat waves that we experience all over Canada, and especially in BC, where we're located. There was a small town close to us that almost entirely burnt down. And these extreme heat waves are not only faced in Canada, but also the rest of the world, as many of you might be experiencing. I know Europe had a bunch of record-breaking temperatures in the last month or two, and our city, Vancouver, is currently in a heat wave as well right now, and it's affecting the world's food supply, especially wheat, rice, maize, and all these super important grains to food security. And we're focusing on these 4 sustainable development goals as well to go with that, including zero hunger, taking action against climate change, and responsible consumption and production. And the plot on the left shows the trend of the rising temperatures in the useful farmland we have globally. And heat stress results in a broad range of changes to the plant cell environment, or to the crop plant cell environment, resulting in a decreased yield as well as a decrease of the nutritional value of the yield we're getting. And this results from a decrease in grain filling during production, a decrease in the photosynthesis synthetic rate, to a downregulation of important genetic pathways.
So then our approach this year is focused on wheat, because it does account for about 23% of British Columbia's crops, as well as 30% of world's grain production. And just keeping in mind that Canada is also the 5th largest producer of wheat, we really wanted to focus on this project as it's very close to home, but also has a large impact globally as well. So our system is to design a heat inducible promoter that will be able to express our genetic constructs to be able to express these enzymes that will be helpful in mitigating heat stress. So yeah, as I mentioned, Canada is especially poised to help develop this agricultural biotechnology and to progress it further, especially with us. The stigma around the world about genetically modified organisms and food, and as a team, we're also helping to kind of reduce that as well, and to understand where the public perceptions are at. So just moving on quickly to provide a snapshot of where our different subteams are at the moment. So for Wet Lab, our present genetic circuitry work is described by 3 different systems. So we have glycine betaine, ACC deaminase, and SBPAs. And the goal of these 3 is to achieve the goal of controlled abiotic heat stress and to be able to manage it. And so here we have ACC deaminase that catalyzes the breakdown of the substrate ACC, which is a precursor to the biosynthesis of ethylene, which is a stress hormone in plants. And for SBPAs, we have it in our construct because of it's a Calvin cycle enzyme that's central to carbon fixation, and it helps in the photosynthetic capacity of plants as well. And then furthermore, glycine betaine on the side here is an osmolyte that's directly upregulated in the action of choline dehydrogenase, and ultimately helps act as a chaperone protein to help fold different proteins in the plant. And so our goal this year is to use these enzyme pathways to offset the metabolic payload required to kind of maintain overall plant health in times of high heat. So we're planning to use protoplasts as our proof of concept system. And our main goal this year is to be able to isolate protoplasts, which are basically the leaf cells without the cell wall, and isolate protoplasts from wheat seedlings to be able to use those to express our gene constructs. And so far, our team has managed to grow many different batches of wheat seedlings. We've gone through a couple of rounds of trial and error in learning how to work with plants, because this is also a first plant project for our team. And we are just at the point of transfecting them, about to transfect them with the constructs to be able to conduct our experiments and test whether our constructs are heat inducible and move further. So I'm moving on to human practices.
Yeah. So for integrated human practices, we started off with kind of evaluating if there is a need for a project, or how it would be conceived by actual farmers around us in British Columbia. So we made a visit to 2 different farms, both on kind of the Vancouver Island area. And the first one, the Sannington farm, which is pictured on the left there, kind of reassured us that there was a great need for a new sustainable method for crop production with the rising temperatures, as irrigation, they were saying, is currently, or more irrigation, is currently the popular solution to growing wheat and with the rising temperatures. And that's quite wasteful or not even plausible for a lot of farms without the system already implemented there. And Fieldside Farms kind of taught us about how heat actually affects their crops, not only from a molecular standpoint, but with what kind of businesses they can sell to. For example, in the summers with or in harvesting cycles with a higher temperature, the protein levels, I think, end up being a lot higher, which makes the crop not viable for like flour kind of beef, but for a beer production or anything like, or something like that instead. And from a wet lab perspective, when we were deciding to use, or when we decided to use protoplasts, we wanted to see if this was a good idea or even plausible. So we consulted with the iGEM Plant Committee, as well as Dr. Song from our campus here. And also to kind of generate or gauge when we were contacting farms and our different contacts, we noticed there was a bit of a hesitancy to accept the idea of GMO or genetically modified wheat as a solution to the rising climate. So we've consulted with Dr. Korolewski, who is in the faculty of the Animal Welfare Program, and Silver Reel Farms to kind of get their ideas on GMO as a solution to rising climates. And that has prompted our idea for the GMO discussion panel, which is one of the panels we're focusing on. It's called the Let's Talk GMO panel, and it's actually happening next Wednesday, as well as the International Accessibility to Synthetic Biology panel happening mid-August. And then to or Pranny, if you want to talk about the community garden food harvest project initiative, because that's something you're focusing on there.
Yeah, for sure. So just to kind of get more of real life experience with working with plants, and also to kind of understand how on the other side of the scale, like organic farming works and like small scale gardening, we chose to help out with our community garden in harvesting food. And as our focus is on food security and reducing hunger, we collaborated with this program locally, and they're helping to harvest this food and distribute it to people who may not have access to enough food. So that's a really great initiative as well. And then just moving on to quickly give a brief snapshot of Dry Lab as well. So at this moment, we are, to kind of better understand how abiotic stress conditions affect wheat, we analyze publicly available data from NCBI using RNA sequencing or RNA-seq data of 1-week-old wheat seedlings that were subjected to different kinds of drought stress, heat stress, and also a combination of both. And so the graph here kind of shows the differentially expressed genes in heat stress wheat as well as drought stress wheat, and then here those that were not significant between the two. So it's really interesting, and we're hoping to use some of this insight to focus on in wet lab as well to kind of inform our assays and experiments. And then for mathematical modeling, our goal is to focus on looking at the reaction kinetics of the expression of our gene circuit and how the binding to the heat inducible promoter happens, and to be able to better optimize it as well. And then also we're focusing on looking at climate modeling in different areas of British Columbia and Canada to be able to look at areas that are more susceptible to heat domes and areas that would more benefit from this kind of a solution. And then for hardware, our goal is, and this is a very future goal, is to design and test a portable device that can be used to detect heat stress in plants just by looking at the wheat leaves, and then also help us detect the activation of our gene circuits as well. So a couple of different kind of where we are at with our different sub-teams, but that was all. And then thank you guys for listening. And if you have any questions, you can also let us know. We're more than happy to answer.
Okay, awesome. I think next we have iGEM Suni Anyanta to present.
All right, that's me. Just give me a second to get my presentation. Okay, there we go. So, hello, I am Derek Yashinsky. I'm one of the members of Team Suni Anyanta here in New York. And I just want to show you what has been going on with our team for our project Cyanospector. So at the beginning of this year, our team started by figuring out where exactly we want to go with cyanobacteria. And initially, what we were thinking of was because of like in local areas, we see a lot of algal blooms that end up shutting down public access to lakes and ponds. We thought that we would try to do something that would help kill cyanobacteria to make it more safe. But after going into more research with how synthetic biologists are working with cyanobacteria, we thought it would be a better idea to do something that would simply take the cyanobacteria from these bodies of water and use them for something more useful. And since cyanobacteria already has viruses that only infect their specific strains, cyanophages, we decided we want to go somewhere down that line. So what we end up deciding on was building a toolkit that would consist of preserved cyanophages that we would engineer them so that there is no DNA or replication machinery inside the capsid. This would allow us to send it to other research teams so that they can insert their own DNA and replication machinery for whatever purpose they see fit. And for our process so far in the lab, we have been using Golden Gate assembly, and we have been just for this over the summer, we have been focusing on level 0 cloning, which has involved us just replicating the genes for the cyanophages. The cyanophage I should mention is STIP37, and we are using E. coli as a host because of convenience. And the genes that we are cloning are the head, neck, and tail of the cyanophage. We are also making a modification to the head of the cyanophage by adding a biotin tag onto it. And the purpose of this tag is so that teams can use a stepped admin plate to pretty much anchor them together. And what they can do from there is, if this is in a lab, they can use this interaction to immobilize sound bacteria that teams are working with. Or if a team is looking for wild samples of cyanobacteria, they can use these plates to collect the bacteria. And finally, another part of our project is reaching out to the public to teach them more about cyanophages, because we felt that the public, especially now, only really sees viruses as a very dangerous thing and don't realize the benefits that genetic engineering presents. So what we decided to do was, at our college, at our the building where our chem department is located, we have a facility known as the Science Discovery Center, and it's pretty much, you can kind of think of it as like a children's museum, where it has a lot of interactable little museum exhibits that teach people something. It's open to the public almost all year round. And what we were thinking is creating our own exhibits to show the public, along with making small assembly kits that we can mail out to other people. And it would basically have people build different parts of the phage, and we have this nice little video here that shows how our little model would be put together. And inside of the heads, we would be able to have like a little model of DNA to represent what research teams are doing right now. And that's it.
Awesome, that was a really cool project. Okay, last but not least, we have iGEM Hopkins, if you guys would like to share your screen.
Oh, can everyone see my slides? Sweet. So hi everyone, we are iGEM Hopkins. This is the second year our team is doing iGEM. The team of Hopkins kind of disappeared in 2016, and 2020 there was an idea to bring it back, and we competed in the 2021 competition, didn't do particularly well, but we're trying much harder, and I think we have a really good project idea for 2022. So there are some really key issues preventing the growth of plants in space. Most importantly, there are no gravitropic cues for directional growth of roots that we have on Earth. Those are absent in space. And so as you can see in the figures on the right, during the period that the plants were actually in microgravity, the roots started growing in sort of a wayward direction. And other papers have sort of replicated these and also found that the effect can be pretty disastrous, as in the roots sometimes even grow in the same directions as the stems, or they just have overall lower plant mass, lower root mass as well. And that's unfortunate, because in the application of space, that would translate to reduced calories for astronauts on missions. So we want to avoid that. And essentially, our project is trying to engineer a replacement for the innate gravitropism pathway with instead magnetotropism, so that we can directionally grow plant roots and hopefully restore some of those losses. A bit of background on how gravitropism usually works within plants. It usually depends on the motion of these large, heavy plastids containing starch called statoliths within the cells of the root meristem, most specifically the columella region. These sink and cause the motion of these pin proteins or auxin carriers. These associate to the statoliths and to the inner membrane of the plants. And what essentially happens is when the statoliths move, the distribution of the pin proteins also changes. Since those are auxin carriers, it causes intracellular changes in the distribution of auxins within the root tip. And eventually, it causes a pretty significant shift in the center of where the auxins are most concentrated, called the auxin maximum. And this shift actually causes a lot of changes in how the elongation of plant cells occurs on either side of the root tip, causing the entire root tip to steer in the direction of the force due to gravity. Now, our sort of engineering attempts to take advantage of that mechanism. So we are trying to load these gravity sensing organelles, the statoliths, with a lot of ferritin, specifically Pyrococcus furiosus ferritin. And then we're trying to look into iron toxicity and other iron tolerance mechanisms so that we can determine what the ideal iron concentration is to load the ferritin and essentially make these statoliths basically susceptible to gradient magnetic fields. We're also including in our design a couple different markers for positive and negative selection, Ruby and neomycin phosphotransferase 2, which I believe confers resistance to kanamycin and neomycin. And this year, instead of using a PCR-based cloning method, we're electing to use golden braid assembly since our genes are quite large. We thought it would be most suited to the eventual destination vector we want to use, which is the Agrobacterium binary vector PLX. Our hardware project is sort of designed to augment, allow us to do a lot of these experiments. So the first thing we have to do is obviously, because there is no microgravity on Earth, and obviously sending projects up to the International Space Station or into orbit is extremely expensive and has a much longer timeline than an iGEM project. We have to simulate microgravity, and so we use a device called a clinostat or random positioning machine. It essentially continuously rotates, and the effect of that is that the contents of the cells never have any time to settle, and the gravity vector becomes averaged in a bunch of different directions. And so essentially for the cells or the plants within the clinostat, they experience simulated microgravity. So that's really useful for testing our device. The other sort of part of the project, which is equally or maybe is slightly less difficult compared to the wet lab engineering, is actually design of a constant current coil driver. That's required to generate a gradient magnetic field that's required to actually move the statoliths. And the reason we went for all this complexity is that we thought making a uniform gradient field or to create a uniform force due to magnetic field was really important for getting the intended effect. So there's a lot of added complexity, included a custom circuit board and some custom embedded firmware.
Okay, so for the dry lab aspects of our project, the first modeling goal that we addressed was to understand how the ferritin that we're introducing will interact with the native ferritin. So we used AlphaFold to look at the docking and concluded that they would interact. And right now what we're working on is a simulation of iron loading kinetics and looking at the like time dependent and then also concentration dependent like equilibrium points. Currently we're using a one compartment model. So basically we're looking at only the kinetics once the iron is inside the statolith. And we're just like limiting it with Michaela's mentioned kinetics, but we are also planning to compare that to our experimental data and then try multi-compartment models to like better understand what factors control the actual time-dependent kinetics of iron loading. And the goal of this would be to compare it to like iron poisoning experiment, and then also beyond the concentrations we test to look at simulated kinetics for concentrations that we don't have the opportunity to test, to look at like the optimal sort of concentrations to use in conditions. And for human practices, since our project is intended to be used in space, there's some sort of connections that we've been making to topics that are large on Earth. And we're sort of reaching out to people in those fields to see how we can translate the understanding of these main three focus areas to space. So for environment and energy costs, we're looking into, well, actually the main one is that space launches are very expensive. So looking at the energy costs for that and the current energy costs for launching space food or water and other resources, and seeing how our project can sort of work with current developments in that area in a responsible way. And then also like in food health and nutrition, how our project can sort of improve astronaut nutrition. So, you know, we've been considering like, because we're introducing more iron into the plants, how that will affect the nutritional value, and then also the ability to translate the project to other types of plants with different nutritional contents. And we've also been interested in agricultural biotechnology and the ethics and policy surrounding that. So like food security.
Yeah, so that's essentially our project for this year. Yeah, that's all we have to show you guys.
Great, that's amazing. So right now what we're going to do is that you already had some time and space to add some questions and comments to the Miro board. So please, I would like to ask everyone to keep updating that throughout the day, or even if you have any questions further, you can always use the Slack channel. We wanted to use the opportunity that all of you are here so that we can have some interaction. So what we're going to do is that we're going to go through the order of the people that have represented, starting with ASU. So if someone has any questions or any comments or something that you want to say, please feel free to unmute yourself and let's see what we can do. So any question for ASU?
Oh, there's one that's on the Miro board right now, and it says, how is the complex targeted for transport to the vacuoles? I'm assuming they're talking about the phytochelatin complex. And then how do you test that the complex ends up in the vacuoles? So this is something that we've kind of been working on right now, Gabby, if you want to talk about it.
Yeah, so it's a bit hard to explain like verbally, but the mechanism of phytochelatins entering the vacuole and complexing with heavy metals is something that's a natural occurrence within a lot of different plants, and also microalgae, specifically the microalgae we're working with too. So this is an already existing mechanism of heavy metal uptake and detoxification within these organisms, but by upregulating this pathway and targeting the specific enzymes that are limited in the pathway, we can increase, hopefully, the ability of this microalgae to sequester heavy metals from water. But regarding actually determining whether that complex has entered the vacuole is a bit complex, because if you remember, I don't think I explained it when I was presenting, but for the proteins that we are producing in the cell phytochelatin synthase and arsenate reductase, we do have those tagged, so we can visualize them via Western blotting. But the issue is, as you might know, phytochelatin synthase is the enzyme that produces the phytochelatins, and the phytochelatins are the things that bind to the arsenite. So, and that's ultimately going to end up in the vacuole. So, it becomes hard to visualize whether the phytokeratins themselves are in the vacuole, because the thing we have tagged is just the enzyme that produces it in the cytoplasm. So that's kind of where the difficulty comes in, because it's easy for us to visualize what we have tagged in the cytoplasm, but not so easy for us to visualize what's in the vacuole. And we were originally thinking about using something called immunocytochemistry to visualize the phytokeratins in the vacuole, but that would require tagging of our produced proteins, which we didn't tag because we only have the enzyme that produces them. So that is a difficulty we're honestly still dealing with in our project. And I think it's something important to think about whenever anyone's doing a project, like how are we going to validate that our mechanism is actually occurring? And that's something we're currently trying to think through, but a lot of the things we can use to validate our mechanism will be more complicated, but I guess that's like kind of a half answer. Like, as with any other iGEM team, we're still struggling with the best way to like visualize and confirm like that our pathway is really happening.
Okay, cool. I think that was all the questions that were for our team, but we can move on to UBCO now. There's a couple of questions on the Miro board already. So the first one is, the person says this might be only relevant for lights near water, but do you have any containment strategies in mind aside from containment inside of the device?
Sure. We haven't explored many other options. Our first idea is to go for an auxotrophic control. Right now, cyanobacteria have been used for a while, so the main control that they have is using knocking out some genes. The one that we are looking at right now is to knock out copper channel, so that it will require us to include much more copper in their medium, much more copper that you will find in any kind of water source or human naturally occurring water source. So if anything happens, they are likely to die. And also something that happens with these organisms is that they are usually organisms that are modified, they're not very good at competitive, competing ecologically. So they are very likely to be wiped out, but still this biosafety measure is what we are taking into account.
I have kind of a follow-up. Is there any concern about like introducing copper in the medium, like in terms of like the cost? I know copper is one of like the earth abundant metals like iron and like nickel and stuff, but have you like thought about that?
Yes, actually, not only because of that, but also because we try to be as ecological as possible. And even if we are working with GMOs, the idea of probably using our cyanobacteria, the liquid, as a fertilizer will be interesting, and having that much copper out is quite concerning. So we are looking for other ways of controlling the auxotrophic control. So yeah, they're definitely going for trying to look for other options.
Okay, awesome. And the second question that's on the Miro board is, are you integrating several bioluminescent genes into one organism, or having a mixed culture of bacteria with different bioluminescent genes?
I can take this one. So we are integrating the entire bioluminescent metabolic pathway into a single organism, Synechocystis 6803. And the bioluminescent genes, there are 5 of them, and we are assembling them into a single construct through Golden Gate assembly. And the genes, 2 of them convert the precursor caffeic acid into the substrate luciferin, one of which catalyzes the light-emitting reaction itself, the luciferase. Another recycles the product of that reaction back into the precursor caffeic acid. And then there's a 5th one that's required for a post-translational modification of one of the other enzymes in the pathway. And like I said, we're integrating all of those into a single organism.
Yeah. Adding to that, we are also receiving a strain from the University of Copenhagen that they already managed to produce caffeic acid in this strain. So our goal is to modify that one as well to truly make a system that is entirely autobioluminescent. So yeah.
Okay, cool. I think Thessaly went next. So one of the questions that's on your Miro board is there any risk of accidental environmental contamination in your project, or is that taken care of the plant's ability to uptake and detoxify contaminants in the water? And it looks like you've addressed this on Miro, but if you just want to reiterate your answer here.
Okay, can you hear me now?
Yes, yeah.
Okay, so yeah, I try to answer this question in the Miro board, but I don't know who made this question. I would like some clarification on maybe on the what do you mean contamination, because we talked about toxins and phosphorus, and these are things that we are trying to get rid of from the environment. But if you're talking about contamination by genetically engineered organisms, like the plants that we are planning to modify, yes, there is also a mechanical aspect of biocontainment, because it will be our system will be enclosed in a kind of mesh, both below the water and above the water. So both seeds and the roots cannot escape into the natural environment. And we're also planning to try and express the Barnes gene in the reeds seeds. So wherever the anthers are developed in the life of the reed, they will be destroyed. And for the next question, what issues have we encountered in working with our chassis? Yeah, we actually have not worked with our plants yet, because we are in the cloning step of the project. We're still assembling the level 1 plasmids, but so far we have not encountered any problem with growing them, because they are used in the laboratory that we are working in. And so, yeah, if we get in any trouble, we will let you know.
Okay, awesome. Yeah. I think the HKU team went next. So the question that is on your Miro board is, why chlorella over other microalgae?
So yeah, why chlorella is pretty, like, there's a couple really easy points. I'll add on to what my teammate also wrote on the board. So the first one is that it's the chlorella strain we have, Autotrophica, was already like localized and found from Hong Kong, which means not only is it already known to work well in our environment, but it's also very relevant to us. The second one is that based on what my friend wrote, it's very, it has capabilities to fold proteins that cannot be folded in like bacterial chassis and yeast chassis, for example. And that capability is obviously useful for scaling beyond just ants. And the last one is also its resilience and its productivity. So chlorella has a very thick cell wall that allows it to be very resilient to a lot of physiological stress, including temperature, pH, physical abuse, which we know personally. And then it's also very productive, obviously. So as I mentioned before, it has a lot of expression capability. It can express proteins very quickly without having to deal with silencing in some situations. And then it's also very productive in the way that it can reproduce very quickly as well. Also, adding on to this, it does have its own antimicrobial effect. Interestingly enough, this is something we've actually seen in our own labs. So this also limits the amount of antibiotics we actually need to use in the lab. So yeah, that's some of the reasons we chose chlorella.
Awesome, nice. Now moving on to UBC iGEM. You have two questions. The first one is, what challenges did you face when first working with RNA sequencing, and can you explain your learning curve and where you are now a little bit more?
Yeah, I just want to start off by saying that I'm not in dry lab, so I'm not probably the best person to answer this question, but just kind of overall from what we've been talking about in our team is that one of the biggest problems working in our project with like RNA sequencing and collecting that data is that like wheat or like T. estivum is not really an organism found in many or is not supported by many different libraries or databases. So it's like it's difficult to find information for like our specific application as compared to maybe like Arabidopsis or E. coli or something like that. So definitely that has been a challenge to kind of focus on our specific organism of interest. And then also kind of considering that the wheat genome is a polyploid, so it's like it has a lot of different genomes in itself. So we've been trying to kind of get help from like experts and trying to filter down which genes we should really be focusing on. And so our team has like conducted an enrichment analysis to look for, as you mentioned, like an overlap between the differentially expressed genes in heat stress versus non-heat stress plants, and just to kind of look at whether those genes include our genes of interest as well, and also to kind of get a like list of, for example, the top differentially expressed genes, and to be able to use that to validate our heat stress model in wet lab as well. But yeah, so we're just at the moment applying more stringent kind of filtering to be able to hone down on certain genes that we to kind of make more sense of that RNA-seq data, because right now we had many, like thousands of genes that are differentially expressed, but we're trying to filter in and hone in on the ones that we're specifically interested in or are actually, and filter out the low confidence ones as well. So that has been a bit of a challenge.
Okay, interesting. That's cool. Someone else asked another question. Are there any consequences of ACC deaminase activity or disabling ethylene signaling?
Yeah, so also with some of the bioinformatics analysis, we're currently looking at the pathways and how the downregulation of ACC or how downregulating ethylene activity impacts other different pathways and metabolism in the plant. But what we know of right now is that ACC deaminase breaks down ethylene, which is a big stress signaling hormone in plants. And ethylene, high levels of ethylene itself leads to stunted growth and low yield. So our aim with this project is to basically increase that growth and yield in plants by reducing ethylene. And usually what happens in high levels of ethylene is also that you have a lot of abscission in like leaves. We have leaves falling off as they kind of further develop, or ripening of fruits as well. So I think maybe one of the consequences of this could be hindering fruit ripening, but as we're focusing more on wheat and not really flower or fruit growing organisms, I feel like that is maybe not as relevant, but we're still kind of in the process of understanding how changing that activity impacts different metabolic activities in the plant. But yeah, that was a really good question.
Awesome. So now let's move on to Sunyanyanta. I think this is a really cool question. So what resources, software, or equipment did you utilize to design and produce your plasmid assembly kit and the toolkit that you mentioned in your presentation?
So for the model that I showed you, I unfortunately was not the person who worked on the model. We actually had someone outside the team work on the model for us, and I'm not sure what software he used. So unfortunately for that, I don't have that information. But for the equipment, we just used a regular old 3D printer, which actually the Science Discovery Center that I mentioned in my presentation actually has, and has for like a open access.
Okay, cool. And yeah, for like any teams that maybe want to like follow up with more information after the fact or like have any other cool resources to share, the Slack is always available for that purpose. Okay, yeah, so iGEM Hopkins has a couple of questions on their Miro board as well. So the first is, why did you decide on using P. furiosus ferritin specifically in your project?
I can take that one. So basically, we just came across this paper describing this point mutated ferritin in P. furiosus that had a really large iron core. So the thought is that for each ferritin particle we express, we would be able to store more iron and hopefully induce more of a magnetic effect in our root.
Okay, cool. Yeah, that makes sense. So this is the second question is also one that I had. Where did you get inspiration for your project idea from, and have there been any other attempts to grow plants in space, and what are the problems associated with that process?
So in terms of, I can have Srini talk more about the inspiration for the magnetropism, but so there have been plants grown in space. It's a pretty exciting field. So the ESA has this project called the Melissa Project, where they work a lot on trying to incorporate plants into like bioremediative strategies. And NASA has a hydroponic system called ponds, which was actually specifically designed to try to solve the problem of poor root circulation. So basically the fundamental problem is that the roots are so terrible that there's like no surface area for them to exchange nutrients with the media. Ponds is basically like a liquid medium that they're growing in that is constantly circulating to try to increase exchange. But that is still limited, probably because the roots are simply, they have low surface area to begin with, and then they're also not properly extending into the rest of the media. So they're really susceptible to like bubbles, for example. So yeah. Srini, do you want to take the first half of that question?
Absolutely, yeah. So as far as the inspiration for the project idea goes, some members of our team are also part of another design team, kind of situated in the same sort of organization, which is the biomedical engineering department at JHU. And that design team is specifically looking into designing a sort of magnetogenetics system as an alternative to optogenetics. And so we were thinking, okay, part of our team has really good experience or some experience with plant engineering. Other part of the team is working on this magnetogenetics design project. And we also have access to the applied physics lab, which does research in space and health and in also synthetic biology. So we thought, okay, my friend and I were just walking down the street. We're like, huh, what if we could put all these ideas together? And I don't know, somehow I just came up with, you know, what if we tried to grow plants in space using magnets? And I don't know, that's kind of, I just put it together into a proposal and pitched it to our advisors and the rest of the team, and yeah, took off from there.
That's awesome. It seems like that's how all the best ideas are made, or just like off the cuff. But yeah, I mean, I think generally, like, thank you everyone for sharing all of your pitches, and it's so cool to see like how many different Phototroph projects are happening this year and how they're all kind of in their own spot, and that just goes to show like how much diversity you can have when you work with phototrophs. But now, kind of as we wrap up a little bit, I think our plan was to kind of just talk a little bit just amongst ourselves about what we want from being part of this community. So earlier, we kind of told you what our plans were in terms of like the handbook, the conference, and then also like the protocol video series. But if anyone has any questions about those three things, or like even our future meetups, now would be like a great time to ask that. And then also beyond that, if anyone has any feedback for us regarding our plans for this year, like maybe say you want a couple more meetups, or you think the meetup should be at a different time, or like anything about that stuff, definitely let us know. So this is just time for us to talk about your thoughts on the community and also what you want to see.
If you also have any other comments or questions in general, there's also space in Meetup if you want to brainstorm a bit. Yeah.
I guess maybe we can break the question down a little bit. I guess we can start with the handbook. So is everyone kind of on board with the handbook and video series idea? And is anyone have any ideas on how maybe we can integrate both of those parts together? Is everyone on board with like knowing what the handbook is, and has everyone had a chance to take a look at it? Anyone have any questions regarding that?
Have we thought about doing more of an interactive handbook? I don't know if I think the last year's one was more of a PDF, but maybe we can kind of incorporate the videos to complement the written portions of the handbook as well.
Yeah,
to kind of maybe have like a video for every protocol or every kind of general feedback that's mentioned in the handbook. So
yeah, absolutely. I think that's a good idea, and a good way of organizing it is just to have a video for each section that kind of like requires some sort of additional explanation maybe. But yeah, definitely.
Yeah, and also adding on, maybe you could like design a website or some sort, and then you can have like the section of the handbook and the video series like together. And that might facilitate like understanding and stuff like that.
Yeah, actually, we really want to do that. Sorry. Also in the discussions, they were going to say that some of us were thinking maybe we should try to put this into some kind of embody it somehow, because right now it's just a document. So maybe a website, something. With any luck, we can even make iGEM integrated. Who knows?
Yeah, I was gonna say a similar thing, like put it on the iGEM website, actually, like René was talking about earlier.
I guess a format that works well for software, I guess, sort of handbooks and stuff like that is read the docs. And I think the iGEM distribution kit GitHub links their read the docs for this year. So maybe that would be an effective platform to keep everything really organized and also enable future updates.
Oh, did you guys also want to talk about your like animation idea? Because I remember you mentioned that briefly when we were emailing, but I thought that was a really cool thing that you brought up.
Yeah, absolutely. I think Cheryl has more experience on doing the actual animations than I do. But the brief idea is there's a decent number of people on our team who are pretty interested in 2D or 3D animation. And so, you know, especially in the context of doing synthetic biology education, I think last year we worked with MIT and made this animation about, I think, type 2S assembly. So any sorts of small topics like those could easily turn into like an animation. And that might help the handbook out as well.
Awesome. Yeah, that would be really cool.
Yeah. Actually, we noticed that there's like a lack of more Moclo sections in the actual Phototroph a bit, especially in the section of the Golden Gate. So maybe that's really cool.
Okay, cool. Does anyone have any other questions maybe about our other meetups that are happening soon, which we will talk about in a second, and or also the conference that we have planned?
Oh yeah, just a slight request because Asian time zone. Would it be like all right if we like push the meetup time like slightly earlier? This is like currently 1:30 a.m. in Hong Kong. So
yeah, sorry, it's really hard to coordinate international timings, but I think for our next meetup, what we can do is we can maybe send out some sort of communication beforehand where everyone can let us know what times they're available and what times work for them.
yeah, sure, that would be great. Thank you so much.
Yeah, it's a bit hard because we have teams literally from both sides of the world. We are on the very west and we have the very east.
Okay, Gabby, I think you can go to the next slide. So here, I don't know if we have the link for it ready, but we do have like a sign-up sheet ready for everyone to kind of just indicate what parts of this collaboration they want to participate in, because obviously it's not required that you do all three. But just so we can get a feel for who wants to do what, we'll be sending out the link to this sign-up sheet soon. It'll also be in the Slack if you need to look at it after the fact. So if you could all just fill that out within the next few days or weeks, that would be really helpful for us. And then, Gus, do you want to talk about our next meeting?
Sure. So again, we are trying to make this a community. So don't forget to join the Slack. Probably we're going to share the link again. And the idea is that now after this meeting that we kind of got used the opportunity to meet each other, we'll be able to put in the Slack what we're going to do. And the next meeting will be something that get a bit more casual. And what we want to do now is that now that we know what our projects are kind of what they are, we would like to ask you maybe in the Slack, try to share your any kind of problems that you had, any kind of things that you're facing or any other things. There's a lot of people in that Slack, not only us. We have previous iGEM teams. We have experts that have already are in the field, both in industry and researchers. So please use those resources to ask questions, even if they seem simple, we can all benefit. Just as a personal experience from BCO iGEM, it's the first time that we are using cyanobacteria. None of our PIs have worked with them in the past, so we have to reach a lot of people. So having this tool is really cool. So next meetup, we're planning on having this space for having a more of a troubleshoot section and having even more feedback. We know that all of us are still working on our projects, so we hope to see your updates. We really want to know what you're doing. This is gonna be pretty soon, in about 2 weeks. So without further ado, just saying that have all of that available. And final remarks, I hope you enjoy all of this. Please be active in the Slack. And again, I hope you're really pumped up for this opportunity, and we hope that we can make this a really cool community for all of us.
And before anybody goes, one last thing is we would like to take a couple of screenshots of the meeting, if that's okay with everyone, just to keep as a record for human practices and all that. So if you're comfortable, feel free to turn your cameras on.
I love these people.
Ryan, are you gonna do a countdown?
I can, sure. How do, sure, yeah. Okay. Everyone. Okay. 3, 2, 1. Sorry. One more time. 3, 2, 1. Okay.
Cool. Thank you, everyone.
Thank you so much, guys. We'll see you in 2 weeks.
See you then.
Thank you.
You can probably stop the recording now.